Okay, let's jump right into this. Welcome to our explainer on a topic that is fundamentally rewriting our understanding of biology and regenerative medicine. For the longest time, we thought cells communicated mostly through direct contact, or just by releasing free-floating molecules into the void. But it turns out, our cells are constantly sending each other microscopic, heavily armored packages packed with complex biological instructions. Today, we're diving into microvesicles, the tiny giants of cellular communication. Here is our roadmap for today. We're going to start with the discovery of these secret messengers, break down exactly how they're manufactured by the cell, explore the tech we use to capture them, and finally, look at these staggering human clinical trials using them to heal the body. So, let's kick things off with Section 1, the body's secret messengers. Now, while extracellular vesicles sound like brand new, cutting-edge science, researchers have actually been building up to this for decades. Back in 1967, scientists observed what they dismissively called platelet dust. They literally thought it was just cellular debris. Garbage, basically. Actually, it wasn't until 1983 that researchers actively observed cells deliberately releasing these vesicles. The field steadily gained momentum as we realized these weren't trash bags, but absolutely crucial messengers, culminating in a Nobel Prize in 2013 for discovering the machinery that regulates all this vesicle trafficking. The crucial starting point here is understanding exactly what we're talking about, because the terminology gets kind of confusing. The broad umbrella term is extracellular vesicles, or EVs, but they fall into three classical categories based on their size and origin. First up, exosomes. These are the smallest, 30 to 150 nanometers, and they're formed deep inside the cell, in endosomes. Then you have our main focus today, microvesicles. These are larger, 100 to 1,000 nanometers, and they bud directly off the cell's outer surface. Finally, apoptotic bodies, the largest ones, which are basically just the fragmented remains of a dying cell. Getting this specific distinction right is absolutely vital for everything we're about to cover. All right, moving to Section 2, how microvesicles are made. Let's look at the biogenesis and shedding. Picture the architectural differences in this process. A microvesicle literally sheds. It pushes out and pinches off directly from the plasma membrane, taking a literal piece of the cell's outer skin with it. Contrast that with exosomes, which are packaged inside a multivesicular body deep inside the cell before being spat out. Because microvesicles bud directly from the surface, their membrane composition closely mirrors the cell they came from. That makes them highly specific to their parent cell's current real-time state. Following that full life cycle, once a microvesicle breaks free from its parent cell, it's basically on a road trip. It travels through the extracellular space, or the bloodstream, to find its target. When it arrives, the vesicle can bind to surface receptors to trigger an immediate signal or, and this is wild, it can be taken up whole, swallowed by the target cell through endocytosis, where it eventually pops open and releases its contents inside. This isn't random floating, this is directed, deliberate, biological communication. But as we dig deeper, we find that microvesicles are not just one single flavor. They are highly heterogeneous. You have your classical MVs, sure, but recent discoveries show we also have ARMMs, a much smaller subclass driven by specific ARRDC1 proteins. Then in cancer pathology, we see large oncosomes. These are massive vesicles that aggressive tumors use to prep distant organs for metastasis. And then there are stress-induced MVs, which cells spit out like SOS flares during low oxygen or inflammation, loaded with warning signals for the rest of the body. So mechanistically, how does a cell actually push one of these out? It requires some insanely precise molecular machinery. Step 1. An activation signal causes a rush of calcium ions into the cell, which wakes up an enzyme called calpain. Step 2, calpain acts like microscopic molecular scissors, cutting the internal protein skeleton that usually holds the cell membrane rigid. At the same time, lipid scramblers flip a molecule called phosphatidylserine to the outer membrane. Step 3, small GD pieces like Rho A and RF6 flex like a muscle. They contract the cellular skeleton to push the membrane outward and pinch it off. It's a highly orchestrated biological dance. Which brings us to Section 3, unpacking the biological cargo. Why do cells go through all this effort in the first place?
Well, they do it because of what's tucked inside. The sheer diversity of functions here is incredible. Once released, these vesicles act like biological FedEx packages. Depending on their specific cargo, they can trigger immune regulation, promote angiogenesis, which is building new blood vessels for wound healing, or even alter a cell's entire metabolism. Because they carry such a massive variety of instructions, they are the ultimate multi-tool for maintaining the body's balance, and for us, a total goldmine for therapeutic applications. Breaking this cargo down by class, we see four main buckets. Proteins, like cytokines, act as immediate distress or immune signals. Lipids help the vesicle physically fuse with the target cell. But the absolute game changer here, the nucleic acids, specifically mRNA and microRNA. When a microvesicle delivers RNA, it is literally handing the target cell brand new genetic blueprints, telling it to start or stop producing specific proteins. This is genetic reprogramming happening naturally in your body every single second. Finally, metabolites like ATP provide the immediate battery power needed for all these reactions. All right, section 4, capturing the tiny giants. If we want to use these things as medicines, we first have to catch them. Isolating these invisible nanoparticles from a complex fluid like blood is a massive, massive engineering problem. Labs basically rely on a few primary methods. Ultracentrifugation spins the fluid at incredibly high speeds to literally smash the vesicles to the bottom of a tube. Ultrafiltration pushes the fluid through microscopic sieves. Or immunoaffinity, which uses tiny magnetic beads coated with antibodies to grab specific markers right off the vesicle's surface. But there is a massive trade-off here. It's the eternal laboratory struggle. If you want a lot of vesicles, a high yield, using simple filtration, you're going to get a lot of protein garbage mixed in, low purity. If you want ultra-pure vesicles using density gradients or magnetic affinity, you're going to get very, very few of them, and it's going to be incredibly expensive and time-consuming. Solving this specific bottleneck is the #1 hurdle for mass-producing vesicle-based therapeutics today. Let's dive into Section 5, the future of healing. The clinical applications. So where is all this technology actually heading? The absolute clinical workhorse right now is microvesicles derived from mesenchymal stem cells, or MSCs. Look, we've known for a long time that stem cells are great at healing, but transplanting whole live cells is super risky. They can form tumors, cause immune rejection, or even create blockages in blood vessels. But plot twist, we've discovered that the stem cells actually do most of their healing just by releasing microvesicles. By harvesting only the vesicles, we get all the benefits: tissue protection, flipping immune cells from an inflammatory to a healing state, preventing scar tissue, without any of the risks of whole live cells. It is quite literally cell-free cellular therapy. And the clinical data we're seeing, it's staggering. In severe COVID-19 patients with failing lungs, a phase 2 trial of a product called ExoFlow significantly reduced 60-day mortality. For knee osteoarthritis, injections of umbilical cord vesicles actually demonstrated real cartilage regeneration in early human trials. In randomized control trials for severe diabetic foot ulcers, weekly topical application of these vesicles dramatically accelerated wound closure compared to standard care. And for pulmonary fibrosis, patients literally inhaling nebulized vesicles saw improved lung capacity in just a week. Seriously, this isn't science fiction anymore. It is happening in clinics right now. But to keep things balanced, let's look at Section 6, limits and next steps, the hurdles we still have to overcome. Because before these become standard treatments at your local pharmacy, we've got major biological and logistical hurdles to clear. As we saw earlier, natural yield is super low, and the vesicles are a highly mixed bag, very heterogeneous. While they're great at healing, their natural targeting ability is kind of limited. If we inject them into the bloodstream, how do we guarantee they all go to the damaged heart and not, say, the liver? Plus, we don't fully understand their pharmacokinetics yet, how fast the body clears them out, and what the exact dosing regimen should be. And finally, they're biologically fragile. They require strict, expensive, ultra-low temperature storage just to survive transit. Okay, let's do a complete lecture recap covering the absolute must-memorize concepts from today. Extracellular vesicles act as the body's ultimate biological FedEx system. A highly likely exam or discussion question on this topic would be, what is the primary architectural difference between an exosome and a microvesicle? The answer you need to lock in. Exosomes are formed deep inside the cell in endosomes before being released, whereas microvesicles bud directly off the outer plasma membrane. Another huge one is, why are MSC-derived microvesicles considered a safer alternative to traditional stem cell therapy? The key answer is that they provide the regenerative benefits, like transferring microRNA for tissue repair, without the risks of whole, live cells, like tumor formation or immune rejection. Also, definitely memorize the 3 biogenesis steps for microvesicles: calcium influx, calcaine cleaving the cytoskeleton, and GTP-cis driving the budding. Mastering these mechanisms is absolutely crucial for understanding where cell-free therapy is headed. Which leaves us with this final thought.
Right now, scientists are actively learning how to open these tiny biological packages, empty them out, load them up with custom-designed genetic drugs, and attach specific molecular GPS tags to their surfaces. If we master this, if we can perfectly engineer these microscopic messengers to target any specific cell in the human body, what incurable disease will cell-free therapy conquer next? It's a fascinating question, and the answer is being worked on in labs right this second. Thanks for joining me on this explainer. Keep learning, and I'll catch you next time.